The sounds you're listening to were generated from data. Data that came from a recording of a teeny tiny pocket of space between two brain cells. This music represents findings about how brains work that have wide-ranging implications for the field of neuroscience, from understanding neuropsychiatric conditions like autism to building human-computer interfaces. So, how do you get a song out of a neuron? And what's it telling us?
Oh, this neuron's really excited.
Simón's son studies neuroscience in the Chen lab at NYU Langone. We paid a visit to the lab, a kind of synaptic recording studio, to gather the data that will eventually end up in that song we just played. Simón studies neuroplasticity, the brain's ability to change and adapt. She does this by taking recordings of synapses, which are the electrochemical connections between neurons, or brain cells.
So if two neurons are connected to each other by a synapse, the one that is sending the signal will spike and then release some neurotransmitters or chemicals that get received by the other neuron.
These cells in the brain can be plastic or change in a number of ways.
One of the ways that we classically think about neurons is that they can exhibit something that we call Hebbian plasticity, where neurons that fire together wire together. And so if two neurons are talking to each other quite a bit, the connection between them gets stronger and stronger.
In other words, Hebbian plasticity is like plugging a microphone into an amplifier and turning it up, strengthening a signal so that it takes less time for information to travel from point A to point B. It's why things that you do frequently become almost automatic with practice. But an unchecked amplifier will just turn up every signal until the speaker blows, which is why we have something called homeostatic plasticity, which stabilizes information in the brain by keeping signals from being amplified too much.
We typically think of these things as two separate processes, that Hebbian plasticity does its thing and homeostatic plasticity comes in to say, hey, keep it down, tone it down a little bit, and keep everything in check.
But Simón is finding it's not that simple.
This idea that they're in opposition to each other is not as clean-cut as we've previously thought.
Simón's experiments are changing the theoretical frameworks used to understand plasticity in the brain, and they could have broad implications for the field of neuroscience. To understand these processes, Simón takes recordings of synaptic activity.
To sort of listen to the synapses that a neuron possesses, I take neurons from the cortex of mice, and I grind them up is a little intense of a word, but essentially dissociate them so they're not connected to each other, and then I take some of them and I put them on a little glass surface, and then they just naturally regrow and form their networks again.
Once they're ready, the dish of neurons goes under a microscope, and a machine keeps warm artificial cerebrospinal fluid flowing so they don't die.
And then after I do that, I take a little electrode and I go right up to one of the neurons. That one looks good. And then I sort of poke it a little bit, and then I essentially get inside the neuron.
Simón then applies a little negative pressure by sucking in some air to attach the pipette to the cell.
Let's see if we can break in.
And then she gives it a little kiss to break in.
Now we're inside. And let's get a recording. Let's see what it's doing. It's almost, I've essentially built a forest, right? I've taken a bunch of little seedlings, I've let the forest grow, and then I go up to one of the trees and try and figure out how it interacts with all of the other trees around it. And from that, I can see all of the different, the electrical responses, the synaptic responses, and use that to answer questions about how homeostatic plasticity works.
Simón's last step is to actually listen to the different electrical responses.
The first time I encounter encounter the data, it kind of gives me that same feeling, right? When you're done with the experiment and you have the data in front of you, you don't know what it's going to say. When I, plug it into the music software and hit play for the first time, it's like a fresh new experience of, what's the result? What am I seeing?
What is she seeing? A recording, any recording, is just a continuous measurement of how something changes over time. When a microphone picks up a sound wave, that's being translated into a voltage, which creates that squiggle you see on a screen. Brain signals are recorded on the computer in the same way. The electrochemical changes are translated into a voltage, and you see a squiggle like you would in a piece of audio software.
If you remember, we were recording from inside a neuron, and so what we're doing here is we're listening to the different synapses that it all has. So it's receiving thousands of synapses from, all of the neurons around it.
The height of each of these responses represents the strength of the synaptic signal. Simón wrote a computer program that takes the strength or the height of each response and assigns it a specific note. Put all those spikes in sequence, and you get this.
It's essentially, you're kind of like hearing all of the synapses pinging.
What's more, neurons that behave differently can sound different. To induce a homeostatic response, Simón treats neurons with drugs that prevent them from transmitting, producing a unique song.
So this neuron was treated with a drug that turns off the spiking, so it can't spike and transmit anymore. And so after a long period of time, there's the homeostatic response where it increases the strength of the synapses. And so the range of the notes in the musical version of this is much wider, and there are more of them.
What Simón is finding doesn't just make for a new hit single. Among other things, it could change how we understand neuropsychiatric conditions.
There's an idea that in autism, that the homeostatic plasticity or the mechanisms of homeostatic plasticity are altered in a particular way. Understanding exactly how a mutation that is associated with this kind of thing changes the way that a neuron acts and is connected with another one could potentially give us some understanding into exactly how autism can come from particular changes to these kinds of proteins in the brain.
There's also evidence that brain recordings can be used to engineer audio that can have clinical applications and even therapeutic effects on people. Research shows that music based on recorded brain signals from EEGs can influence people's breathing rates and make them more relaxed by creating a personalized tempo. Scientists are using this same technology to construct brain-computer interfaces that convert brain signals of people who have loss of motor control into music. But for Simón, music is just another context for data, a way to differently communicate the ongoing chatter of the brain.
So this is another way of giving the data a voice and allowing it to speak to us in the same way that when we're analyzing it and trying to find different kinds of patterns in it. No. No, let's not go with that one.